Hey, everybody. Good morning, good evening, good afternoon, depending on where you're joining us in the world. Welcome to the MechE Alliance seminar series today with Felice Frankel. Felice is a photographer, an expert, I'll call her visual communicator. As a research scientist in chemical engineering, along with support from mechanical engineering. And amongst her fabulous work, she's been awarded a Guggenheim Fellowship. She's published a number of books addressing visual communication. And today she's going to engage us in a conversation. And the question she wants to ask is, are your tools and timing biasing your data? Felice, thank you very much for joining us today. And I'll turn the screen over to you. Participants, please feel free to chat your questions, Q&A, and we'll hold them for Felice until the end. But please,
Wonderful.
The floor is yours.
Terrific. Thank you so much for having me. I hope it's fun, and maybe hopefully to get you to think a little bit, because I'm thinking about this a lot. I'm going to share my screen. Okay. Yes, this is something that I'd like to encourage you to think about, because I've been thinking about it. The interesting thing that happened when I submitted a title to the group, which, by the way, thank you so much, Brian and everybody, Teresa, Irina, for inviting me. It's great to be with you guys. So this is the initial title, which is a little long, but it was basically, it's some thoughts on informational bias when visually communicating science and engineering. It's something I've been thinking about, which I'll get to in a moment. And as anything else, from one experience, it gets you to thinking about something else, in the sense that when I got the email back, this sounds like a good title, the email said it sounds timely. Now, it just happens at that time when this was being arranged, there was a great deal of conversation going around about racial bias, which of course is an extraordinarily important topic. But the point of my bringing this up is that because the word bias was used in newspapers, television, all over, specifically addressing racial bias, I got to realize once again that something is informing our thinking. The fact that this was a conversation that was taking place biased the approach to my title. I hope I'm explaining that right. So in a way, I changed it to, are your tools and timing biasing your data, to clarify it. But after that email, I basically thought, you know what else? Not only are your tools and timing informing your thinking and your data, but your preconceptions are. My point is that because we were in that state of defining bias in a certain way, that's how that word was read. So I hope I'm clarifying this whole notion of what I mean by bias. Merriam-Webster defines it in this way. It's a systematic error introduced into sampling or testing by selecting or encouraging one outcome or answer over others. That's what I'd like to talk about for you today. Now, here's the issue. I am a photographer. I have never written one line of code, but I do work with computer scientists to talk about their visualizations. It is coming from, frankly, a very naive point of view, in the sense of my not truly digging deep into collecting data. So just keep that in mind. But my hope is that what I'm going to talk to you about, even though most of them are pictorial, will be somewhat metaphoric if you are collecting data. But once again, I've always started my talks to encouraging you to understand that any sort of representation, that is with data or visualization of photography or graphics, they are representations. And that when you gather it and when you decide to display it, it requires you to make decisions. That is the place where I think bias can, in fact, get in the way. Again, maybe I'm showing my insecurity to you computer scientists, but I want you to consider what I'm going to show you as relevant to your own decision making. So the first issue with perhaps informing your data is, what tools shall I use? It's not trivial. I've seen it in my own work. For example, here you see a picture that I made quite a while ago of Toyo Tanaka's work. And these are gels that have absorbed some fluorescing dye. That's good enough for this conversation. I was using film at the time. That's spelled F-I-L-M. Maybe some of you have heard of it. It turns out that this particular kind of film, Fuji film, did not capture a certain wavelength. It did not capture orange. And this is the image that I got. That was the tool that I used. The tool was film, this kind of film. What my eye saw was this image. You see that there is orange over here. This is what my eye was seeing. So when I digitally created the image from the film, I fixed these particular rods into orange. In this situation, I'm suggesting that the manipulated image is more honest, but the manipulated image only took place because I was able to see where the orange wavelength was. So my perception of the material biased me, but the bias in a way became more real or more honest. By the way, when I had a discussion with the editors at Nature, I was putting this in a previous book, they felt that my manipulation was not allowed. That even though that was the real image or the real information, they felt that my coloring would not be permitted, even if I indicated, which I always do when I'm doing anything for publication. They felt it was not permitted. I think that we could argue that fact, but I'm getting to the point here of showing you that my preconception, which was an honest preconception, made a better data, because these images are basically considered data. Now, again, getting back to the point of the tool, this is an image that I made quite a while ago as well on a microscope, which enabled me to adjust the lighting. These are yeast colonies from Jerry Fink's lab. And just by changing the light source, we get a completely different picture. And we could ask different questions about this picture. For example, in this image, I'm not interested in what's going on in this empty space because I'm not seeing it very clearly. In this image, because the tool was slightly changed, I am seeing material or structure that was not clear in the first one. And so the tool is informing what I am seeing. The idea of what is your point of view will bias or affect or inform what it is that you're seeing and what it is that you will report. So again, I'm showing you old images because I've been thinking about this, that it's been in my mind for a while. This is a picture I made an eternity ago. I used to be a landscape architectural photographer. This was an artist's installation in a field in California. And it was a series of poles. And from this vantage point, this is the image that I made from this point of view. I changed my point of view later in the day, walking around the place, and I found that the poles were ordered. They were not as random as I initially saw, because that initial point of view is different from this point of view. Again, I'm encouraging you to relate these photographs as metaphors to your own points of view. What about the time frame? When are you collecting your data? It's something that I think needs to be considered more often, frankly. And once again, my metaphor and reality for me is, for example, again, out of my former life as a landscape photographer, I took this photograph of a winery in California. This was in the dead of winter. Tried to stay in the identical spot 6 months later, and I'm getting a completely different image in every way. I'm going to put these side by side. So I think I actually did a pretty good job of placing myself in the right spot. I was kind of surprised at that. But yes, this is about photography, but at the same time, it relates to all of our data. Depending upon when you collect it, you'll get different data, probably. And this, reminding you of the how we alerted you to the talk, this is one image of a 1 centimeter sandwich, a glass sandwich of block copolymers that when the hexane evaporated, it changed colors. The block copolymers, you could see the change of color. This was taken 24 hours later, which is the same material. And this is the series that I made of, like, I think I took it every 3 minutes. And you see it starting from here, going to here, and eventually going to the last one. And so depending upon when you collect your data, you're going to, you're biasing your material. Here's one that I'm clumping together with a few. What should I include in my collection? This is a decision that you have to make. How much of the what should I include? And what shall I leave out? These are all about how much, and they're all separate. So there is a reason why I'm including them all in your thinking. So for example, in this image, I made, these are yeast colonies. And I talked the researchers, Jerry Fink, to grow the colonies together in a Petri dish, which is not what you would normally do, but I wanted to show in one image the comparison of the morphologies of the colonies. They differed with their genetic component, and so they showed hyphae, and the wild type did not show hyphae. And so, but I didn't like this image because I felt that my process was too much a part of the image. You see the light source in the back. And so what I did was I zoomed in more, and I am now showing you less of an image that is less stuff, but I feel that you now are seeing more of it because I'm editing it down to what I'm trying for you to make a comparison about. And so in my decision, I decided to edit it down because I wanted you to see certain structure. But if that's not what I wanted you to see, and just look at the whole line of colonies, then that decision was not a good decision. It's the same thing with what you are going to eventually, hopefully, visualize with your data, what to include and what not to include. Here's something that used to be shown quite a while ago to supposedly show the process of evolution. Now we're talking about what to include and what not to include. Well, this is dead wrong. First of all, we didn't necessarily all become white men, but the most important part of this mistake in this editing is that they're not including the extinction growth, which is all about what evolution is about, that some things do change into different species, but they don't make it. It's survival of the fittest. They didn't make it. And so they deleted very, very important information. Okay, you can argue it's just a diagram or illustration, but I'm trying to get you to think of the importance of including what you think you should not necessarily include. In this example, for example, you see here a micrograph that I made, and you could see a reflection that I felt it was in the way. This is a chip of some sort. And so I digitally deleted that reflection because I wanted you to see the other data, let's use that word. And in this case, I think it was an okay thing to do. I considered that reflection as an outlier. Now, however, in this case, you're seeing something called Proteus colonies. I made this picture. I think it blew me away to see that these colonies are growing in this amazing pattern. And it was for a book called On the Surface of Things with George Whitesides. But you see that there are cracks in the agar. And I wanted the reader to only pay attention to the patterns because that's what the image was about. So watch your screen as I digitally deleted the cracks. Whenever I do anything like that, I always indicate that. I'm not an artist, I'm a science photographer, and I would not do this, for example, for a journal article. I would do it for a cover. But in this conversation, I'm talking about how much should you include when you are biasing your data. And I'm raising the following question. By deleting these cracks, I am deleting what I considered were outliers. By deleting the outliers, I'm informing what you are going to see, which was a decision that I made. Was I permitted to do that? Should I do that? Am I biasing my data, which is in fact the picture of the Proteus colonies? So the question I raise for you when you are working with your own outliers, are some outliers more important than others? And if that is the case, if we delete these outliers, are we deleting important information? So here is an example that it was published all over the place because it was quite remarkable how David Desrosiers put together over 100 SEMs to create this composite of a flagellum. Now, importantly, as I'm reading from the article, this composite electron micrograph of the flagellum body and hook produced by rotational averaging, I have to close this because it's overlaying, the motor proteins and export apparatus do not survive the extraction procedure. I'm showing you this again. The motor proteins do not survive the extraction procedure and so are not shown. So this researcher considered the motor proteins as outliers to make this image to appear this way. that's what he decided to do. In the end, because this image got so much attention and it looks so incredibly perfect, it was published all over the place, including people who believe in an intelligent designer, and replicated with illustration that only an intelligent designer is going to create something so perfect. And that intelligent design is a better explanation than chance. So by deleting all the protein, the motor proteins, you are informing and biasing the image. And it's an issue, it's a question. So I just, again, I'm talking photographically, but I really do believe that it's connected to any kinds of data that you're collecting. So how should we decide on these outliers? I mean, I'm raising questions for you. I have no answers, but I'd love to talk to you about it. And moreover, can we teach the machine to decide on what outliers we can delete? Because I'm getting into what everybody is thinking about now in machine learning. I'm not sure, this is something I'd love to talk to you about. Can we teach the machine? Are we biased in our teaching? If we're going to give only a certain collection of material for the machine to learn from, are we already biasing what the machine is learning because we are telling it what to teach itself? One of my most naive questions that I ask my computer science friends is, if all of this can happen, can we make our systems mutable? So for example, when I was talking to somebody who is teaching the machine to look for cancer cells, particular cancer cells, so that when the machine looks at another slide, it will identify that cancer cell. What if there's a cancer that we don't know about yet, and we haven't yet thought about teaching the machine to look for that cancer cell that we're not clear about? Is there a way to teach the machine to look for it without ever showing it in the first place? Can the machine teach itself to look for something that it has no information about? I don't think so, but I'd love to hear from you about that. So I'd love to hear from you. It was short purposefully because I would so much prefer having a conversation with you about this. And once again, although I'm showing you photographs, these are data, and you are all working with data. And I'd love you to consider thinking about this stuff. So that's it. And I'd love to talk more if we can.
Felice, thank you. I send you the virtual and the physical applause here. So participants, please go ahead and enter your questions in the Q&A or the chat. But Felice, let me just take a chair prerogative here. And I think any researcher on the call is very familiar with reading a paper and then going and trying to replicate that experiment, that result, and saying, yeah, there's something missing, and go talk to the researcher and say, oh yeah, I did this under the half moon with this particular fork that had a particular, and so there's all these details that are potentially missing. Often, that not intentional, but just sort of because it didn't seem important, or it was too much information contextual to capture. So that's sort of the first comment. Second comment is, at the same time, I'm personally seeing a lot of, at least startup journals trying to do more like video journals, where you have your written sort of article, but coming in with the video camera to try to capture the full experimental process, which presumably would help to capture some of those things that it's too easy to forget in text. So I don't exactly have a question, but sort of more that's the sort of things that I was thinking of when through your presentation, the fact that we naturally have to leave things out in the written form and the photograph form, and maybe just wanted to get your perspective and sort of some of these other media for publishing, such as video journals, to try to capture the full context and situational awareness when performing an experiment.
Yeah, it's a wonderful thing you're raising, and my understanding, certainly Nature, for example, asks for original data or visualizations, and with no manipulation, for example. They ask, and they're supposedly able to test whether what you're showing has been processed. I can discuss whether I think we could get around all that. But the bottom line is, I think the more you talk about how you acquire your data and your photographs, the better off we as researchers will be. And more important, for me, more important, because my whole thing is talking to the public, the more people understand how we get our stuff, I think the more involved they will be in science. I think that's just presenting what we get is not going to cut getting the public involved with what we do. I think it's critical. I think that's one of the biggest problems we have and continue to have with COVID. People still don't understand what this is all about because we're not learning how to speak to people. I think one way, and this is why I'm working on these handbooks, I think one way of getting people engaged is by showing them how we do what we do. I don't have data on this, but I am convinced that the process is just as interesting as the final product and the thinking behind it. In fact, right now I'm in the middle of gathering examples of iterations with illustrators, how you started with this beginning point and where you wound up with the final point, and the process, your thinking process of why you changed the color and what did you leave out. That's fascinating and gets us more involved with the science itself. There should be sections on this. And it doesn't have to be video, by the way. It can be just good still images showing your lighting, showing your DPI, all those things that are the technical parts of making your images and collecting your numbers. It should be part of it. I agree with you. Yeah.
Thank you for that. So participants, I have a question here that I'll relay, but please go ahead and enter your questions in the Q&A or the chat. So a question that was texted to me or chatted here. I think it's asking really sort of in terms of the complexity of how to so there's information that we choose or to convey and not, but just I guess the question is around the time.
Yeah.
So the trade-off and sort of capturing all this context data and the process vis-a-vis getting the material out. Again, I don't think there's an easy answer there, but
no, well, I don't know. That's why I'm giving you this opportunity to talk, because I will tell you that I gave a little bit of this talk. I was invited to speak to some Microsoft executives, and I brought these ideas up. And after the talk, quite a number of people came over. You're raising some very interesting questions, certainly about time. And then they said, but we don't have time to do this. We just want to collect the data. Thank you very much, but no thank you, basically, is what they, and so I don't know the answer, but again, if you guys are so good at creating the algorithms to collect the data, have you thought about collecting the data at various time frames and making those comparisons? Of course you have. In some fields, somebody's doing that. But is it worth it? For that's the bigger question. I'm trying to get you to see it's worth it, because you're basically changing your perception by not looking at the whole picture. But is it possible to look at the whole picture? And especially if that whole picture constantly changes. I don't know the answer to that. I mean, I'd love to hear from somebody out there who is, do you consider time when you collect your data? Is there a way to do that? Is anyone doing that now? Anyone want to just raise their hand and talk? No, huh? Your sound is off, Brian.
Oops, sorry.
Okay.
Go ahead and chat your questions in the Q&A. I think we have the raise hand function turned on. So if I don't.
That's correct. If you raise your hand,
okay.
we can unmic or invite you to talk.
So is someone out there when they're collecting their data, are they thinking about the time frame? Is specifically the question. And if you're not, I guess I'd like to know why not. Because maybe the thing that you're collecting your data about doesn't change. Maybe you're looking at structure, for example. Although if you're in biology, everything changes all the time. So how can we teach the machine to correct itself if something is changing over time? Nobody's up.
One of the questions that I would add to sort of build on that, there's a Q&A here. Somebody asked the question, can you define time frame a bit more?
Very good. Oh my God, that's wonderful. You bet. No. I mean, it all depends. Of course, sometimes you're working in picoseconds. For me, I was working in 6 months. But I'm wondering if you are just looking at femtoseconds, or are you missing something? Probably that gets a little crazy, but if you're looking at molecular interactions, then you have to look at that scale. But maybe by taking a step back and looking more in microseconds, that will give you some information that you're not getting if you're looking at femtoseconds. So I don't know what time frame. I say all of the above. Of course, this is easy for me because I don't have to write the code for that. So, but it's a great question. It depends. It all depends. I'm trying to suggest that the larger comment I'm trying to say is we all get stuck in a tunneled vision. I do too. And that we might be missing important things about what we're seeing if we don't occasionally. I mean, you have to write your PhD, so you can't do that, but just occasionally take the 30,000-foot look, and maybe you can see something that will inform you in a different way.
Vivek, you have your hand raised. Teresa, are you able to unmute and?
Yes, you should be able to unmute now, Dr. Sudhir.
Yeah, so can you hear me?
Yes, we hear you.
Yeah, thanks, Felice. That was a wonderful talk. So I want to take you up on this question of time and give you maybe a couple of perspectives that came to my mind. So I work in precision measurements where we try to study extremely tiny effects, which mean that we are forced to convince ourselves that we are not fooling us when we try to measure some small effect here or there. So indeed, there is a very rigorous attempt to try to replicate experiments over multiple times. Different temperatures in the lab, different conditions that might happen outside the lab environment, all sorts of other things that we don't actually know about or have control about. Having said that, I do think that, as a scientist, there is always a pragmatic approach to this question, in the sense that I feel like the job of a scientist is to falsify certain hypotheses. So, perhaps you conduct an experiment, see something, and then maybe you have a hypothesis that, well, the thing that you just saw might disappear tonight. So, you go in and see whether it disappears. If it doesn't disappear, well, you come up with a new conjecture, which says that maybe the effect disappears next week. But of course, this cannot be continued indefinitely, and that's the other point that I wanted to make. Because imagine that you're really in the habit of, let's say, studying a single apple, And of course, if you want to see whether the apple exists, here and is red today versus here and red next week, of course it won't be, because by next week it would have started rotting, and by next year there's no apple. So you're no longer studying an apple; you're really studying the environment in which the apple used to be after a year. So I'm not sure I understand this open-ended question of yours, where you say that keep measuring something for an arbitrary long time. My answer is even the universe doesn't exist for an arbitrary long time. So that doesn't actually hold water.
No, I can't. How could I conceivably argue with you about that? Of course you're right. You can't. There's no way that you're going to look at something at every time frame. I mean, it's just. I guess it's. Let me put it this way. I don't know how to work an SEM. Whenever I make SEM images, I work with a technician or a scientist who knows the ins and outs of SEM. But occasionally I will make an SEM to see if I'm missing something structurally. And it's just something that I have in the back of my mind without really understanding the serious mechanisms of how electrons can do what they do in scanning electron microscopy. I just want to suggest that you're not going to study every moment in time. Just think about it, because there might be, through the years of your research, you might start thinking, I wonder what is going to happen if I look at this at another time frame. I'm not naive enough to think that you're going to be able to do it. I'm just raising the issue of keeping it in the back of your mind. And I can't possibly argue with you that you shouldn't bother, because you can't do it. And you've got to get your degrees, and you've got to get grants, and you've got to show the results. But it's just something to keep in mind. That's all I'm suggesting. And maybe keeping it in mind will encourage you to think differently at a particular time frame. That's about all I could say.
Thank you for that, Felice.
Okay.
So I think we have one more question here. It was chatted to us. I'm more of a scientist than an artist, but like to cultivate my creative eye further to try to capture images like the ones you've shown, Felice. What is your advice beyond what, sort of think about it? Then,
So now I'm not an artist, but what I do is I use artistic tools, let's say. I'm not trained at all, by the way, in photography. This is all self-taught, just as you all are self-taught. Let's be real. And if you really have an interest in kind of upping the quality of your images, you should do two things. You should buy my book, which is called Picturing Science and Engineering. And you should also, seriously, because honestly, I don't usually do this, but the series of tutorials that we published on edX and now is on OCW, they're really, really good. They're tutorials on how to, believe it or not, use a flatbed scanner to make some amazing pictures of your work if it's optical. Obviously, if you're starting to get into suboptical, that's a whole different ballgame. But on OCW, it's called Making Science and Engineering Pictures. And the course is not being run now, but the tutorials are. And honestly, they're really very highly produced. You don't see me, it's only voiceover, and all you see is what is going on in photography. So I'd like to suggest that you take a look at those two things. There, I did it.
No, that's perfectly, and I agree, Felice. It's phenomenal material. So I do encourage people to go take a look. So I encourage everybody to continue the conversation. Felice, thank you so much for your time today, and for your interesting perspectives and for your questions. And panelists, thank you for your, or attendees, thank you for your time. So next week, or in 2 weeks, we'll be taking off. It's a busy time of the year with end of semester and some finals. And then our next talk will be June 16th. And I'll be sharing along with Jose Pacheco and Dave Hart a little bit about the experiment that we are endeavoring on in the department to explore further the intersection of data and compute in mechanical engineering, in particular in design and manufacturing. And in this coming year, we're launching a pilot activity that will allow us, through the context of our Master of Engineering in Design and Manufacturing degree, collaborate with companies and alumni with projects that have a stronger data and compute focus than maybe we would have in the past. So we'll be talking more about that on June 16th. Until then, this is our regular channel to engage with you, the community, to invite our photographers, our students, our investigators, and our alumni and company partners to discuss and present the things that are on your mind. So if you have recommendations for yourself or for others, please drop us a note. And with that, Felice, again, thank you very much for your time today, and everybody stay safe and be well, and see you in several weeks. Thank you.